Hello, everyone. Welcome to a new lecture of our course on processing in memory systems. Today, we continue talking about the use case of a real processing in memory system, and we are going to focus on bioinformatics applications and in particular on genome sequence alignment. This work that we are going to present has been recently published at the Bioinformatics Journal with the title A Framework for High-Throughput Sequence Alignment Using Real Processing in Memory Systems. There are two archive versions. One is shorter, the other one is longer, that you can find at the bottom of the slide, and the source code is also publicly available. Let's start with a brief executive summary. First of all, genome sequence alignment, as we will see, is typically memory-bound on conventional processor-centric systems like CPUs and GPUs, and processing in memory, as we know, has the potential to overcome this data movement bottleneck. In this work and in this lecture, we present alignment in memory, a PIM framework for genome sequence alignment on general purpose PIM systems. The alignment in memory framework supports multiple alignment algorithms like Needleman-Bunch, Smith-Waterman, GenASM, and the WFA algorithm, and its first version has been implemented for the PIM architecture. In our evaluation, we will see that sequence alignment on PIM is really promising and has the potential to be significantly faster than on CPU and GPUs. This is an outline of this presentation. We will start with some background and motivation about sequence alignment on CPU. Next, we will briefly describe the processing in memory system that we use in this work. And next, we will talk about alignment in memory, the framework for sequence alignment on PIM systems. Finally, we will go with the evaluation. So let's start with the background and motivation. Genome analysis is the background that we need in order to understand what this work is about. And we start defining what the genome is. A genome is a complete set of an organism genetic instructions, and the DNA is a string of base pairs or characters, basically three different characters, A, C, G, and T. The human genome, for example, has 3.2 billion base pairs. And the goal of genome analysis is to understand genetic variations, predict the presence or an abundance of microbiomes, monitor disease outbreaks, develop personalized medicine, etc. There are sequencing machines that only provide randomized fragments or reads, which need to be mapped to the reference genome in order to make sense out of them. And depending on the sequencing technology, there might be either short, about 50 to 300 base pairs, and long reads that are something around 10 kilo base pairs and 100 kilo base pairs. A key step in the read mapping process is sequence alignment, and this sequence alignment is typically performed using dynamic programming algorithms such as Smith-Waterman or Needleman-Bunch that perform in quadratic time, and they also have quadratic memory requirements. There are more recent, more efficient state-of-the-art algorithms such as GenASM or the WFA algorithm or the WFA-Adaptive algorithm. All of them are supported by the framework that we are going to present today. But in order to give you some background and some understanding about sequence alignment algorithms, we are going to focus on the basic version of Smith-Waterman that performs local sequence alignment. And in this Smith-Waterman algorithm, we compare two sequences, A and B. And for every two elements of A and B, we are going to obtain a similarity score. This Sij will be, for example, 3 or -3, depending on whether the element AI is equal to the element Bj or not. We are also going to consider a gap penalty, that is a linear value. In this case, we are going to consider that this gap penalty at distance 1 is equal to, but if we are at longer distance, it will scale linearly. What's the gap penalty? And in Smith-Waterman, one thing that we'll have to do is computing a scoring matrix. We compute the scoring matrix using a certain expression, like this one is the maximum of the neighboring values and either adding the similarity score or subtracting the gap penalty. For example, if we want to calculate Hij of the scoring matrix, we will have to use this element here and add the similarity score, and then we will also have to use this element here and subtract the gap penalty and use this previous value here and again subtract the gap penalty. After that, we will compare these three with 0 and we will take the maximum that will be the value of Hij. So the first thing that we have to do is to compute the scoring matrix. We are going to assume that we align two sequences, AACC and AATC. And the first thing to do is to calculate this element of the scoring matrix, which is the first element that we have to calculate. And we calculate it by using the neighboring values that we have here and using this expression. Observe that the two elements that we are comparing, A and A, from the two sequences are the same. So that means that the similarity score is equal 3 in this case. And that's why we obtain these 3 for that position of the scoring matrix as a result of the formula that we see on the right-hand side of the slide. Next, when we go to calculate the next element in the scoring matrix, we will have to use these neighboring elements. And again, first of all, will be to compare the two elements of the sequences, again, A and A. So this scoring, the similarity score is again 3. And by using our expression, we will obtain these 3 for that position of the scoring matrix. Next element in the same anti-diagonal, we also need to use the neighboring values to compute it, and we apply the same expression and obtain these 3 here. And we can continue applying the same formula to anti-diagonal by after anti-diagonal and compute the entire scoring matrix. So as we can see, to calculate one anti-diagonal, we need the 2 previous anti-diagonals. In the second step of Smith-Waterman, we will do the traceback operation that is going to give us the exact alignment. One thing that we first do is finding what's the maximum in the bottom right corner of the scoring matrix. In this case is that 7 that you can see on the slide. And that means that the 2 corresponding elements of the sequences are the same. In this case, C and C. Next, we find the maximum, and in this case, we see that there is an insertion because there is a T in one of the sequences, but there is no T in the other sequence, and that changes the direction in the traceback operation, as you can see. And then we continue finding the maximum in this traceback operation. Now we have A, and finally, again, A and A. So for the traceback process, as you see, we basically need the direction of the score calculation. There are other sequence alignment algorithms. We mentioned already Needleman-Bunch. We also mentioned other versions of Smith-Waterman, such as Smith-Waterman-Goto, and more recently presented GNASM and the waveform algorithm. These algorithms have something in common, is that they don't scale well on CPU. If we look at the execution time of these 4 algorithms or 5 algorithms, if we also take into account the adaptive version of WFA, 4 different number of threads on a CPU, we see that the performance tends to saturate as the number of CPU threads grows. So there is no good scaling on current multi-core CPU systems. What's the reason for that? We can observe as well that the instruction per cycle go down as the number of CPU threads grows. So as the number of CPU threads grows, the IPC decreases, and what this basically means is that threads are spending more time being idle. But why are they idle? basically because as we increase the numbers of CPU threads, we'll have more contention in the access to memory, and this will cause more memory stalls for the CPU threads. So as the number of CPU threads grows, threads spend much more time waiting for memory, and that's the main reason why the scalability is limited. How can we alleviate this data movement bottleneck? By using processing in memory techniques. In processing in memory, we can alleviate the data movement bottleneck because instead of accessing data through a narrow funnel, as we typically do in conventional CPUs, that we need to access through the narrow off-chip link, we can access data from the DRAM banks directly. As you see on the right-hand side of the slide, PIM cores have direct access to their own DRAM bank. This could be one processing near memory system, and this way the aggregated bandwidth, the total bandwidth, is going to be much, much higher. So in this work, we are considering, same as in previous lectures, real PIM system with certain characteristics. There is a host processor that has access to standard main memory and PIM-enabled memory. The PIM-enabled memory contains multiple PIM processing elements, and these processing elements typically run at few hundred megahertz and have a small number of registers and small or no cache or scratch pad. And these processing elements need to communicate or may need to communicate via the host processor. This is how one of these processing in memory systems looks like. And in particular, in this work, we have used the ABMEM PIM architecture containing general purpose processing cores called DRAM processing units or DPUs. In this presentation, in this talk, we will use interchangeably the term DPU or the term PIM core to refer to the cores near the memory banks. Each of these PIM cores can run up to 24 PIM threads that are called tasklets, and they support 32-bit integer arithmetic, but they don't support multiplication or division or floating point operations. Each of the DPUs has access to its own DRAM bank that is called MRAM and its own scratchpad that is called WRAM. Observe that in the sequence alignment algorithms, as we have just seen in the example of the Smith-Waterman, we don't need complex operations. Its computation is mostly about comparing different elements from different sequences and also adding or subtracting scores or penalties. So in principle, we don't need to use multiplication or division or floating point. And this is why these algorithms, at least in principle, are very good fit for such a PIM architecture. Now let's talk about alignment in memory or framework for sequence alignment on real PIM systems. As I mentioned earlier, supported algorithms are Needleman-Bunch, Smith-Waterman Goto, GenAthem, and the Wolfram algorithm. And the very first thing that we are going to see is how the framework distributes the workload, the total computation that needs to be done over the different PIM cores and different PIM threads that run in every core, and how they access data. So for the very beginning, we have the CPU thread, the host CPU that reads sequence pairs from an input file and stores them in main memory. And in the next step, the CPU transfers the sequences from the main memory to the MRAM bank, to the DRAM banks in the PIM DIMMs, nearby the PIM cores. Basically, the CPU, the host CPU is going to partition the total amount of sequence pairs evenly, and each of the DPUs will receive the same or more or less the same amount of sequence pairs. In the next step, we will have inside each PIM core, we will have the different threads aligning sequence pairs. And one important consideration in this framework is that the threads, the different PIM threads, are going to work individually on the different alignments. So basically, each PIM core receives a bunch of sequence pairs, and these are further distributed over the available PIM threads, such that different pin threads operate, work completely independently on different alignments, and this way we avoid any sort of inter-thread or inter-pin core synchronization, because it's more of an embarrassingly parallel implementation where each thread can directly operate without having to coordinate to other with other threads or other cores. In the next step, we will have each thread copying a sequence pair from the DRAM bank to the scratchpad using a DMA transfer, and after that, the threads will align the sequence pairs and extract the alignment operations using the traceback operation. Remember that that's the second step of the alignment after having computed the scoring matrix. In step number 5, the threads write the alignment score and operations to the DRAM bank via a DMA write transfer. So after the alignment is done, the result of the alignment will be saved by the thread in the DRAM bank, in the MRAM, that will be later accessible to the host CPU to retrieve the final results. And that's actually what the CPU is going to do in step 6. When the execution on the PIM core finishes, the host CPU can read the alignment score and operations of each sequence pair, and finally, if needed, the CPU will store the alignment results in an output file. Relevant to the implementation of this framework is how we manage data. And this is important for intermediate data structures. For example, the scoring tables that we typically need to use in these dynamic programming algorithms. The scoring table, in principle, or these intermediate data structures, in principle, it could be better to store them in WRAM, basically because WRAM is a scratchpad and it's fast. It's faster than the DRAM bank. It's faster than the MRAM. However, this scratchpad is fast but has limited capacity, and the consequence is that at some point we will, if we want to operate or we want to align larger sequences, we will also need larger intermediate data structures. And because the total amount of WRAM is limited, it might turn out that we will have to reduce the number of concurrent or the number of active pin threads. So we cannot run so many threads in the pipeline, in the core, due to the size of the intermediate data structures. And if we are using less threads, we are wasting cycles in the pipeline, and the overall performance will be lower. So in order to alleviate that situation, we can use another way of managing the intermediate data structures that combines the use of WRAM and MRAM. In this case, the intermediate data structures reside in MRAM, but we are going to use the WRAM as a data cache. So this data cache is in the scratchpad in the WRAM and is loaded on demand. And even though on average we will have longer access latency because than in the WRAM only solution that we presented in the previous slide, because we have to access MRAM from time to time, but the upside is that it will be possible to run more concurrent pin threads, so we will be able to keep the pipeline busy, or at least more busy than in the other case, and hopefully obtain better performance. And one last consideration with respect to the data management is concerns the WFA and the WFA-Adaptive algorithms. And the reason is that in these two algorithms, or in this algorithm and its variation, the intermediate data structures are of varying size. These are the wavefront components, and these requires dynamic memory allocation. The AppMem SDK provides an incremental dynamic memory allocator, but this is shared by all pin threads. And that means that we would need to use synchronization every time that we allocate extra memory. We'll need synchronization for all pin threads running in the same core. Be aware of that dynamic memory allocation. So to avoid this overhead from synchronization, we designed a custom per thread memory allocator for low overhead dynamic memory allocation, and now threads don't need to coordinate to do the dynamic memory allocation. You can find more details in the paper. Now let's go with the evaluation. First of all, our evaluation methodology, we have used real and synthetic data sets for the different experiments, and we have evaluated different systems. First of all, a PIM-based system or a memory-centric computing system, which is an UPMEM-based system with more than 2,500 PIM cores, and then we have compared to 3 CPU systems with different number of cores, different number of threads, and also importantly, different L3 cache size. You can find the details in the table of this slide. So let's start with the evaluation. And first of all, we are going to compare the performance of the different algorithms on the 3 CPU systems. Note that these 2 plots show the execution time for different algorithms, Needleman-Bunch, Smith-Waterman, Goto, Genome, WFA, and WFA-Adaptive for different read lengths from 100 to 1000. And we are also using different edit distance threshold. The edit distance is basically the error between two different sequences, how dissimilar, how different 2 different sequences that we are aligning are. And we or different applications require different amount of dissimilarity, and that's what the threshold defines. So if we are considering edit distance threshold equal to 1%, that means that there might be differences of 1% between the 2 sequences. And one first observation here is that for the different algorithms, the CPU system that has the larger L3 cache is the one that performs the best. And that's an interesting observation because it aligns very well with the fact that these sequence alignment algorithms are memory bound. The larger the L3 cache, we can alleviate a little bit more this memory boundedness. So it makes sense that the system, the CPU system with the larger L3 is the fastest one. One other observation when we compare to the, well, we compared the PIM execution time as well. And in this plot, we are including the CPU to PIM transfers and the PIM to CPU transfers. Remember that we are using a system with a MIM memory and the PIM-enabled memory, and we need to move sequences in the beginning from the main memory to the PIM-enabled memory, and then at the end the results from the alignment results from the PIM-enabled memory to the main memory. The observation here is that the PIM system outperforms the 3 CPU systems in the majority of experiments, and we observe very interesting speedups, more than 4 times for Smith-Waterman Goto, or 2.56 times for WFA-Adaptive. So this clearly shows the big promise of PIM systems for these sequence alignment algorithms. We also show results for PIM execution time excluding CPU PIM and PIM CPU transfers, and here we see even larger speedups, up to 28.14 for the WFA-Adaptive algorithm. And this shows even more promise for the PIM systems. The problem is that in the current PIM system, the current UPMEM PIM system, we really have to move the data from the main memory to the PIM-enabled memory. And that's because PIM-enabled memory is not working as a main memory. In the future, we could expect PIM systems that can either avoid or hide these CPU PIM and PIM CPU transfers by either using the PIM DINs as main memory or, in the worst case, overlapping data transfers and computation. So even if the PIM-enabled memory needs to be a separate memory from the main memory, hopefully we will be able to overlap the data transfers, the copy from main memory to the PIM-enabled memory with computation on the PIM cores, and this way hide the overhead of the data transfers. We also make some observations for the algorithms with regular access patterns. These are Needleman-Bunch and Smith-Waterman Goto, and the one key observation here is that PIM is not able to outperform the CPU for these algorithms when we are using a small read lengths. And there are several potential reasons for these. when we have more regular accesses, the CPU prefetchers can operate nicely, so they can bring to the cache hierarchy the data or the part of the intermediate data structures that are needed. And especially when the read lengths are small, it's more likely that reads and also the intermediate data structures fit in the cache hierarchy, and this alleviates a lot the data movement bottleneck. So this explains why the PIM system is not especially good for these algorithms with regular accesses and when using a small read lengths. We also have more comparison, WFA and WFA-Adaptive for large sequences. One good thing of these algorithms is that they have linear space complexity, and this means that they are really unlikely to exceed the SIMDRAM capacity. And that's why we can go even further and test significantly and experiment with significantly larger read lengths. Like in this plot, you can see read lengths of 5,000 and 10,000 base pairs for different edit distance thresholds. And overall, we see that the PIM system continues to outperform the CPU systems for large read lengths. Only for the longest reads that we have used and also the highest edit distance threshold, we see that the scalability starts being limited, but this is again due to the WRAM capacity. Now the reads are so long, and because the edit distance threshold at least as well is high, we have to keep more wavefront components. And in the end, this is going to be limited by the WRAM capacity. So that's one reason why we don't see further scaling. But in the end, there is also a lot of promise for WFA and WFA-Adaptive on PIM systems, and there are possible software solutions that are out of the scope of this work for even larger sequences. For example, reduce the WRAM footprint by streaming partial wavefront components from the MRAM, or by running multiple PIM threads per alignment. Remember that in this in the current version of alignment in memory, we are assigning one alignment per thread. But one way to alleviate the limitations imposed by the limited WRAM size is to use multiple PIM threads per alignment as well. And hopefully in future PIM systems and in future upman PIM cores, we will see also larger WRAM capacity that will alleviate this limitation as well. In our paper, we also compared PIM to GPU, in particular for WFA and the WFA algorithm. And as we can see in the table, the PIM version outperforms the GPU version in a majority of experiments. And finally, for the intermediate data structures, remember that we have two ways of managing the intermediate data structures, either WRAM only or WRAM plus MRAM. We run some detailed experiments, as you see here, for the 5 different algorithms and 5 different read lengths. We show results for the WRAM only approach and for the WRAM plus MRAM approach. Some observations are that for large data structures such as Needleman-Wunsch and Smith-Waterman-Goto, WRAM only does not scale because the size of the intermediate data structure, the scoring table, is pretty large. So at some point, we won't have so much WRAM to store the intermediate data structures of all PIM threads. So at some point, we have to reduce the number of PIM threads that are running concurrently. Then we observed that for algorithms with small data structures, that's the case of GenASM, WRAM only is better because the data structure is pretty small. We can continue using the WRAM only approach even for relatively long reads up to 1,000. And in this case, we see the best performance for the WRAM only version, and for algorithms that use medium-sized data structures, and that's the case of the two versions of WFA. WRAM only is better for short reads, while WRAM plus MRAM is better for long reads, because they can with this approach, we can run more PIM threads in the same core and make better use of the available resources in the pipeline. You can find all the details about this work in the bioinformatics paper, you have the links to the two archive versions in this slide, and also a link to the source code. We actually have two repositories for the source code. You have both links in this slide. And just to conclude, again, the executive summary, we have seen that genome sequence alignment is typically memory bound. We have seen some motivational results on CPU processing in memory can alleviate the data movement bottleneck for these applications. And that's why we have developed and now present alignment in memory, a PIM framework for genome sequence alignment on general purpose PIM systems. This alignment alignment in memory framework supports multiple alignment algorithms such as NW, SWG, GenASM, and WFA, and it's implemented for the UPMEM PIM architecture. In your evaluation, as we have seen, the main conclusion is that PIM is really promising for sequence alignment and has good potential to be significantly faster than CPUs and GPUs for these algorithms. This is all for today. I hope that you found this lecture interesting, and I hope you found this project interesting. Take a look at the paper if you want to learn more, and don't hesitate to contact us if you want to discuss anything or learn more about the alignment in memory framework. So thank you very much for your attention, and see you in the next lecture of our course on processing in memory systems.